Genetics is a branch of science which deals with study of heredity variation among related organisms. UK established the Institute of the Biotechnology. ICAR International Centre for Genetic Engineering and Biotechnology, which has two centres, India and Italy. When cross is made between two parents differing in two characters known as dihybrid cross. Mendel's first law of segregation. Mendel's second law of independent assortment. When F1 cross with its parent, it is known as back cross. When F2 cross with recessive parent is called as test cross. Monohybrid test cross gives 1:1 phenotype. Dihybrid test cross gives 1:1:1:1 phenotype ratio. The genetics of qualitative phenotype is called as Mendel's genetics. Alternate form gene is known as allele. Albinism is controlled by simple autosomal recessive allele. A gene or locus which masked the action of gene at another locus was termed as epistasis. The gene or locus which was suppressed by an epistatic gene known as hypostatic gene. A gene whose phenotypic effect is sufficiently drastic to kill the bearer is called lethal gene. W.S. Sutton and T. Bovary suggested that chromosomes were the physical structure, which act as a messenger of heredity. The gene is a chemical determiner. The phenomenon of multiple effect of single gene is known as pleiotropism. The work on fish chromosomes began with the study of Ritziat and Katz-Chens. Fish cytologists had been documented chromosomes in India for 125 sp. In metacentric chromosome, centromere located at the center of chromosomes. Acrocentric centromere located at the end of chromosome. In salmonids, satellite chromosomes have been reported. The length of longer arm of the chromosome divided by the length of the shorter arm is known as arm ratio. The arm ratio is always greater than 1. IMC 50 chromosome. Common carp and goldfish, 104 numbers. Heteropneustes fossilis, 56 numbers. C. butrachus, 50 number. C. striatus, 40 numbers. The sex-linked inheritance in fish was first described by John Naismith in Guppy. Gonochoism is the basic mode of reproduction of sexuality. Protogynous, hermaphroditism, female change into male. Protoandrous, male change into female. Synchronous serenity. In XO and XO sex determination system, O is the symbol for number of chromosome. The tendency of gene to retain together during the process of inheritance is known as linkage. Linkage groups in fish were first detected by Winge. Genetic distance is expressed in the units of crossing over or centimorgan, cm. 1 cm is equal to 1% crossing over. Fish is a highly effective and rapid technique for use in gene mapping. Fluorescein isothiocyanate, green color. The phenomenon in which two homologous chromosomes fail to separate is called non-disjunction. A pair of homologous missing from the diploid set known as nullisomic. Trisomic, one extra chromosome. For chemical mutation nitrosoethylurea, NU, are used. In India 44 intergeneric and interspecific hybrids have been produced. B form is present in most DNA in the cell. In chi-square test the probability is less than 0.05 or 5%. Monohybrid ratio 3:1, 1:2:1, 1:1. Dihybrid ratio, 9:3:3:1 or 1:1:1:1. Dominant epistasis, 12:3:1. Recessive epistasis, 9:3:4. Masculination of fish is usually carried out by 17-alpha-methyltestosterone. The aquaculture would expand to 76 million tons in 2030. IMC contribution towards world aquaculture is 4%. IMC contribution towards Indian aquaculture is 84%. Over last 2 decades, Indian fisheries have grown 6.5 times. A chromosome atlas of Indian fishes depicting karyotypes of 128 teleosts found in Indian waters. NBFGR, National Bureau of Fish Genetic Resources. Fish, fluorescent in situ hybridization. In present scenario, conserved mitochondrial DNA genes such as cytochrome B, cytochrome coccidase are used for phylogenetic and taxonomic validation of fish species. There are 50 DNA markers were identified. SNP, single nucleotide polymorphism. EST, expressed sequence tags. Meiosis is the process of primary gametocytes develops into eggs or sperm. Monohalide is a bioactive compound obtained from Pacific sponge. Gene sequence, operon. Sofia, State of Fisheries Aquaculture. Leading country in ornamental fish culture is Singapore. Sterile fish, triploid fish. Number of carbon in progesterone is 21. C banding is the most common type of banding for fish. Reddy et al. Made the first attempt of inducing polyploidy in Roja by using colchicine. A process of adding one or more set of chromosome to the original diploid complement is polyploidy. Allopolyploidy is a process of addition of set of chromosome to the diploid hybrid genome. Sex reversal for male, 17-alpha-methyltestosterone are used. Sex reversal for female, 17-beta-estradiol are used. The process of transferring the foreign gene to the fertilized egg is known as transgenesis. Microinjection is the most common method of introducing transgene into the developing embryo. Genetic drift is the random change in the gene frequency created by the sampling error. The role of microchromosome not fully understood. It contain redundant genetic material. Crossing over increases genetic and phenotypic variance in population. The dominant gene action produce 3 genotype and 2 phenotype. There are 6 types of epistasis are present. Incomplete dominant gene action produce 3 genotype and 3 phenotype. Sex-limited phenotype will express only in one sex. Distance between 2 gene is determined by crossing over percentage. Random change gene frequency is known as genetic drift.